Good morning everyone. Myself Gagan.K of second year second sem College of Horticulture Kolar. Today I will be presenting on biofertilizers. Introduction. A biofertilizer is a substance which contains living microorganisms. When applied to seed, plant surfaces or soil, it colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply of availability of primary nutrients to the host plant. As we know, biofertilizers add nutrients through the natural process of nitrogen fixation or maybe solubilizing phosphorus and stimulating plant growth through the synthesis of growth promoting substances. Now we shall know about what is biofertilizer. Biofertilizers are primarily natural fertilizers that are microbial inoculants of bacteria, algae and fungi separately or may be found in combination. They help in biological nitrogen fixation for the benefit of plants. They build up the soil microflora and thereby bring about good soil health. Biofertilizers also include organic fertilizers like manure, compost, etc. Then coming to the types of biofertilizers, we have biofertilizers containing bacteria, fungal, algae, aquatic fern and earthworms. Then coming to bacteria, the biofertilizers having bacteria. For these examples will be symbiotic nitrogen fixers like Rhizobium, free living nitrogen fixers like Azotobacter, algal biofertilizers like BJ in association with Azolla, phosphate solubilizing bacterium, primarily Pseudomonas, then fungal biofertilizers like WAM, earthworms, etc. Now coming to bacteria biofertilizers, the live cells of bacteria are used as biofertilizers here. The nitrogen fixing bacteria work under two conditions, maybe symbiotically or free living, non-symbiotic. In symbiotic nitrogen fixers, example Rhizobium, the Rhizobium lives in the root hairs of the legumes by forming nodules and they capture the atmospheric nitrogen and provide to the plant. Then coming to Azospirillum, it mainly present in cereal plants. It inhabits both root cells as well as surrounding of roots. It forms symbiotic relations and increases the nitrogen fixing potential of the cereal plant. Then coming to free living bacteria, examples like Azotobacter, Lepsiella, it will not associated with the plant. Azotobacter provides required amount of nitrogen to the plant from the soil. Then coming to these are the steps involved in the mass production of the biofertilizers. These are the flow chart. Then the use of carrier material in production of biofertilizers. The use of ideal carrier material is necessary for the production of good quality biofertilizers as they maintain the health of the biofertilizer. Then coming to the preparation of inoculants packets, these are the steps followed. Then coming to the specification that a polythene bag requires to maintain the quality of the biofertilizers. Then coming to another biofertilizer type like VAM, that is vascular avascular mycorrhiza. The mycorrhiza is a term taken from the Greek language which means fungus root. That is, VAM is a close association between the roots and the fungus. VAM helps in nutrient transfer mainly of phosphorus, zinc and sulfur to the plants. Then coming to the mechanism of action of VAM. The VAM forms an association with plant roots. It penetrates in the root cortex and spreads around the roots of the plant. As the name indicates, they possess sac-like structures called vesicles which stores phosphorus as phospholipids. The other structure called arbuscule helps bringing the distant nutrients to the vesicles and root. Then these are the flowchart for the mass production of VAM. Then coming to the use of they enhances the feeding areas of the plant root as the hyphae spreads around the roots. They mobilize the nutrients from the stems to roots, stores the nutrients as phosphol feeds. They also remove the uh, phenolics, which are the toxic chemicals which otherwise hinder nutrient availability. Then coming to algae as a biofertilizer. Here commonly used biofertilizer is bluefin algae in association with azora. These are mainly used in paddy fields. Then coming to the structure of the BGA. Then Azolla as a biofertilizer. Azolla is a tiny freshwater fern common in ponds, ditches, and rice fields. It is used as a biofertilizer for rice in all major rice growing countries including India, Thailand, Korea, Philippines, Brazil, and West Africa. Then coming to biofertilizers application methods, there are three main methods following, that is primarily seed treatment, root tipping, and soil applications. Coming to seed treatment. Seed treatment is a most common method adopted for all types of inoculant. The seed treatment is effective and economic. Seed treatment with Rhizobium azotobacter azoospermilum along with phosphate solubilizing microorganisms. Here seed treatment can be done with any of two or more bacteria. No side effect is found in seed treatment method. The important thing has the seeds must be coated first with Rhizobium or Azotobacter or Azoospermilum. When each seeds get a layer of above bacteria, then the phosphate solubilizing microorganisms inoculant has to be treated on outer layer of the seeds. This method provides maximum number of population of each bacteria required for better results. Mixing the any of two bacteria and treatment of seed will not provide maximum number of bacteria of individuals. Then next method is root tipping. Here application of azoospermilum with paddy or vegetable plants is followed. Here the required quantity of azoospermilum has to be mixed with 5 to 10 liter of water at one corner of the field, and all the plants have to keep for minimum half an hour before sowing. Then coming to soil application, here phosphate stabilizing microorganism has to be used as a soil application. Here 2 kg of PSM per acre mixed with mixture of PSM with 400 to 600 kg of cow dung along with half bag of rock phosphate if available. The mixture of PSM, cow dung and rock phosphate have to be kept under in tree shade or ceiling for overnight and maintain 50% moisture. Use the mixture as a soil application in rows or during leveling of soil. Next, the precautions to be taken while handling the biofertilizers are: store biofertilizer packets in cool and dry place away from direct sunlight and heat. Use right combination of biofertilizers. Rhizobium is a crop specific, so use in specified crop. Do not mix with chemicals. Use the packet before expiry only on the specified crop by the recommended method. The advantages of biofertilizers: it includes renewable source of nutrients, sustain soil health, supplement chemical fertilizers, replace 25 to 30% chemical fertilizers, increase the grain yields by 10 to 40%, decompose plant residues, and stabilize CN ratio of the soil, improve texture, structure, and water holding capacity of soil. No adverse effect on plant growth and soil fertility. Then coming to some more advantages, that is stimulates plant growth by secreting growth hormones, secrete fungistatic and antibiotic like substances, stabilize and mobilize nutrients, eco-friendly, non-pollutant and cost-effective method. Then coming to disadvantages of biofertilizers, it include biofertilizers require special care for long-term storage because they are alive, must be used before their expiry date. If other microorganisms contaminate the carrier medium or if growers use the wrong strain, they are not as effective. Biofertilizers lose their effectiveness if the soil is too hot or dry. Thank you.